And I want to do a brief introduction, okay?
Sure.
Sure. Welcome everyone to the Ontology Summit 2022. Today is the 9th of February, 2022. And the theme of this summit is pandemics and other disasters. And so we have two speakers today, Chris Mungall and Justin Reese. And I will turn it over now to Ravi to introduce them in more detail. Ravi?
We have great pleasure in welcoming Chris Mungall and Justin to this wonderful series of sessions on pandemic and other disasters. And their topic is also dear to us because we have spent one summit on knowledge graphs. And we are eager to listen to what they have to say about COVID and knowledge graphs. In that regard, I request my co-champion, Gary Bergcross, to introduce our speakers a little bit more in detail. Thank you.
Thank you, Ravi. I am very happy to welcome our two distinguished speakers today to the 2022 Ontology Summit. Both Chris and Justin are from the Lawrence Berkeley National Labs, LBNL, and a fuller introduction to their background is on our website. But briefly, I want to say that we're starting with Chris. Chris Mungall has a PhD from the University of Edinburgh School of Biological Sciences and is a staff scientist in the Environmental Genomics and Systems Biology Division at LBNL, where he heads the Biosystems Data Science Department and where he leads a group working on the application of computational techniques to problems in life sciences of relevance to the health of humans and indeed the health of the entire planet. So very relevant to our topic this year. He described his main interest broadly as, quote, the application of artificial intelligence, knowledge-based methods, and biocuration to the advancement of our understanding of the interconnected roles of genes and the genetic mechanisms in key biological processes. Chris is perhaps most widely known as a force behind the gene ontology, something relevant to today's discussion.
Yeah.
He's also been involved in many of the bio-related ontologies, such as the human phenotype ontology, as well as its disease ontology. Within the ontological field, he has numerous contributions to both ontological methods, ontological design patterns, principles for evaluating the quality and sharing of data and knowledge. A small example is the work on Miro, Guidelines for Minimum Information for Reporting. Last year's summit, we discussed, for example, systemizing definitions in ontologies, an area that Chris has contributed mightily to. Justin Reese is a computer research scientist working on environmental genomics and system biology at Lawrence Berkeley National. He's broadly interested in using computational methods to extract actionable knowledge from the biomedical and biological data, and notes a particular interest in using these methods to address disease. Again, very relevant to our topic this year. 2 years ago, the summit focused on knowledge graphs, as Ravi mentioned. Part of Justin's notable work includes a framework to support customizing knowledge graphs for COVID-19 responses, something we should hear more about today. Justin has a PhD from the University of Virginia in computational biology and bioinformatics. And with that, let me now turn it over to Justin and Chris.
Can I just say thank you so much for that lovely intro, Gary and everyone. It's a great honor to be speaking to the Ontolog group. I've been a member of the list for a while, and even when I don't contribute, I enjoy seeing all of the discussions go by. So Justin's actually going to present this work. It's actually, he envisioned the project and did most of the work. My role these days is mostly keep the money flowing in. But I encourage everyone to just use the chat for questions, and I can field as many questions as I can as we go along, and then we can hopefully have some time for discussion at the end. So thanks again. I'll let you take it away, Justin.
Thanks, everybody. Thanks for giving us a chance to tell you more about this KG COVID-19 project. And Chris, you should feel free to jump in if I forget to mention anything along the way here. Chris is being a little modest about, I think, his contribution to this. He was instrumental in all of this happening. So what I'm going to tell you about is KG COVID-19. That's our knowledge graph for COVID-19 response. Here's just a quick summary of everything we're going to cover. So I'll sort of make the case for why knowledge graphs are a good approach to this, and then tell you about the actual KG COVID-19 knowledge graph. The knowledge graph, I think it's not immodest to say that this KG COVID-19 knowledge graph is sort of morphed. We sort of use this pattern for what I would describe as hopefully a KG ecosystem that we're making called KG Hub, which I think consists of at least 5 or 6 knowledge graph projects. We think it's sort of a useful pattern that we can apply to making KGs for other purposes besides COVID-19. I'll tell you a little bit more about that. But the reason we actually were originally interested in making KG COVID-19 was to do GraphML for drug repurposing. And so I'll describe our work in actually applying GraphML to the knowledge graph that we made, and then some work about how you can actually verify the knowledge that you extract from KGs using GraphML using observational studies. Okay, so I probably don't need to spend too much time convincing you all that knowledge graphs are a sensible way of approaching this problem. But so when we first, this would have been obviously about 2 years ago when we first started thinking about how we could contribute to the COVID-19 response here at LBNL. We were involved in 2 consortia that were started up in response to COVID-19. The one is NVBL, National Virtual Biotechnology Laboratory, which is a consortium of US national labs, including LBNL and RNL and a few others. And we're also involved in N3C, and we continue to be very involved in N3C. This is the National COVID Cohort Collaborative, which is a consortium of US hospital systems that share COVID data and in coordination with NIH and funded by NIH. So when we started thinking about how to actually approach this problem, it's immediately evident that there was plenty of data around. So even before the pandemic hit, you had SARS-CoV, the first one, MERS, and several other coronaviruses, and there's a fair bit of data on all of those even before the COVID-19 pandemic hit. And then, of course, the pandemic hit and everybody started producing scads of data. I mean, you have drug data, viral host protein-protein interactions, sequencing data, Go annotations, epidemiological data. I mean, the list goes on, and we're still, of course, being inundated with data. And there's more of this by the minute. This is a little bit out of date, but at about mid last year, I think there were 23,000 papers, and they were doubling every 20 days about COVID-19. And this isn't really just underscores how much data there is on COVID-19. And so the lack of data certainly in any case is not the problem. Data integration really is the main challenge here. The data about COVID-19 tends to be siloed. It tends to be trapped in texts or PDFs or in publications, or if it's not, it's they're behind some API and separated that way, or in various file formats. And so when we started thinking about how to help here, from our perspective, integration of the data was really key, and it's something that I think we decided we could help with. So the reason for that is if you're going to try to extract some actionable knowledge from all this data, any one use case typically spans a few data sets. So for example, when we started thinking about how to dive into the data, you can, for drug repurposing, for example, you can ask, what drugs are FDA approved? So what do we have to work with that's already been approved? That's pretty easy. You just do a Google search. You ask what proteins are targeted by FDA approved drugs. That's a little harder, but still possibly just download a file and do a grep, or probably even on some website, you can pull out this information. If you want to start digging down a little deeper, now you're talking about a task that involves several data sets. For example, what FDA approved drugs target a human protein that interacts with SARS-CoV-2. So this is sort of getting into toward the question of like what drugs might be good drug repurposing candidates. If you're going to do that, now you're talking about downloading several files. You got to join them in some way. You got to normalize the IDs, and you're probably going to have to write some bespoke ingest code to transform the data to sort of harmonize between these different data sets. And this is really where knowledge graphs sort of shine. So if you go to the trouble of making a knowledge graph that takes all the data in that you're interested in, then what this query that I just described becomes a fairly simple Sparkle or Cipher query. So here I'm showing you the actual results from a query. Here's a SARS-CoV protein NSP8, which interacts with human protein HLA-A, and that interacts with C5. So this is sort of a second order interaction between this protein and this drug. And likewise, here's a second order interaction between the S protein and ribavirin. So this is just an example of a fairly elaborate way to extract data with a fairly simple Sparkle query once you set up the KG. And so I assume everybody here is fairly familiar with what we mean by knowledge graphs, but conceptually this is very simple. We just represent entities that we want to put into the KG as nodes. These are people, places, genes, or diseases, or just any concept that you want to represent. And then the relationships between them are edges. So this is obviously very flexible, and it's very easy to add new data compared to something like a relational database. And it really gives you a pretty rich representation of fairly complex data. So we're not the first people to have the idea, of course, to apply GraphML to KGs. You know, Facebook and Twitter and a lot of companies in the Bay Area where we are use this to great effect to find, for example, among their network of users, who should they introduce to who, and sort of make new connections in their social network. So in the context of KG COVID-19, our idea was to make this knowledge graph and then apply GraphML for drug repurposing. And I'll tell you a little bit more about that later. And after we made the knowledge graph, of course, GraphML is not the only thing you can use these for. So it became clear that other use cases were fairly easy to and useful to do. For example, hypothesis-based querying. There's a lot of scientists here at LBNL who wanted to help, and we can hand this over to them, and they can use it for hypothesis-based querying of drugs that they might be interested in, or different proteins that they may be experts in. We're also using it, this is sort of a work in progress for this year, to integrate clinical data with our KG COVID-19 knowledge graph, which thus far we have not done. So where do ontologies fit into all this? So there's obviously, everybody in this call knows there's a pretty rich set of biomedical ontologies. For example, Go can sort of represent what genes do, and Kebi can represent chemicals. There's the HPO ontology that represents pretty much all of human phenotypes. And then Chris has made this Mondo disease ontology. And so this gives you sort of a high level general sort of modular data that you can drop into a knowledge graph, but it doesn't have any sort of instance level data. It doesn't have any data about what patient has what phenotype, or what proteins have PPI, protein-protein interactions with other proteins. And so knowledge graphs in combination with ontologies really give you an ideal way of sort of integrating all this data, and so the ontologies sort of form this scaffold on which you can put the data that you extracted from all these upstream sources. So we sort of dove in and just spent several months integrating all the data that we thought was relevant for our use case. And this is obviously focused on drugs and biochemical and biological data. There, of course, is a lot of other data, sort of geographic data, and more epidemiological data, or sequence level data that you could put in here that we have not thus far done. But I'll walk you through what we have. So like I said, conceptually, we've sort of built, we've used these biomedical ontologies as kind of the scaffold or the core of this knowledge graph. And that's Go, which gives you gene functions, HPO, that's phenotypes, chemical ontologies, and disease. And so then we've got a lot of drug databases here, TTD, Drug Central, Pharm GKB, Campbell, and Drug Central. So yeah, there it is. Uniprot gives us human proteins and SARS-CoV-2 proteins intact in string or protein-protein interactions. This is the COVID-19, well, I should say the SciBite annotations of the COVID-19 data set. This is basically represents the COVID-19 literature. So you get to know, in this paper, they talk about this gene, and say this drug, and so on and so forth. So it's a way of sort of extracting the data from literature and including it in our knowledge graph. So we have about, well, you can see the numbers here. This is the number of nodes. You got about 60,000 drugs, 50,000 or so publications, basically all the Go terms. Go, 20,000 or so diseases, basically most human genes and proteins, and all the SARS-CoV-2 proteins, obviously, and all the human phenotypes. So these are just two examples, just to give you an idea of the flavor of these edges that are in the KG. Hydrochlorothiazide molecularly interacts with ACE2. ACE2 is mentioned in this publication and so forth. So we have about 24 million edges in this KG. This represents, we think, a useful set of data for drug repurposing in other use cases. So like I said, our intention was to do GraphML in this for drug repurposing, but once we went to the trouble of putting this together, we sort of handed it over or exposed it to NVBL and N3C. Those are the two consortia that we were working with. And this was used, for example, in NVBL to prioritize drug targets. And it was sort of this AIM3 cross-cutting aim from NVBL. And this was sort of integrated into this dashboard that sort of was kind of a clearinghouse that we exposed to LBNL and other researchers that were interested in helping with the COVID-19 response. In the N3C consortium, so N3C has several million now, I think 4 million 4 million patients in there with their COVID-19 related data. And so this is a way of sort of including the public biomedical knowledge for COVID-19 with the case level clinical data in that enclave. We've also done things like semantic similarity calculations for COVID patients using their HPO phenotypes. This is kind of work in progress, and we hope to publish something on that this year. So this is just an example of sort of use cases other than GraphML that have been applied to KG COVID-19. Like I said, well, I won't dwell on this too much, but we aspire, and I think we're getting there, to turn this into sort of a KG ecosystem. And we developed some patterns that we think are broadly useful for constructing KGs and contexts other than COVID-19. So there's 5 or 6 projects now that are sort of in this what I call loose federation of KG projects, and I won't go into all of them, but all this to say, we think that the patterns we develop for COVID-19 or KG COVID-19 are useful generally for making KGs for other use cases. And these are basically, design patterns like a simple ETL platform that leverages some software we've written in our group called KGX and other software that sort of helps construct and manipulate knowledge graph data. And you got other things, obviously, like stable URLs for graph data, which is a bit more challenging than you might think. You get version builds and a lot of downstream tooling that we built to actually manipulate and look at and use the KG after you've made it. Oops. Here's just a brief look of all the tooling we've built downstream. We use this CI/CD software called Jenkins to actually do the builds in a regular version way every month, at least, if not if not more often. This is KGX. This is software for manipulating the KG once it's actually constructed, and it's actually used in construction of the KG also. We put the builds on in a browsable form on S3, and all the artifacts that went into generating the KG, and a subgraph for every component that was transformed and from every upstream source is also available, and a README describing all the artifacts, and the Jenkins file describing exactly how the build was done, and the knowledge graph itself in 3 different formats, and then a dashboard to see what's in it. But this is really the use case that we were interested in, was GraphML, and so we, and in coordination with some Italian colleagues, Luca Capoletti and Tommaso Fontana from Giorgio Valentini's lab, have built some, we think, good GraphML software that I'll tell you about in a minute here. Okay, so now we have the KG built. How do we actually do graph machine learning on this? So we built this sort of meta package called Grape that is comprised of 2 components, in Smallin and in Biggin. And Smallin is the graph library for actually reading in a knowledge graph. And in Biggin, once it's read in Biggin is a graph machine learning library based on Smallin itself, and also TensorFlow. We've implemented a bunch of algorithms. The one that I'm going to tell you about most today is called NodeDevec, but it's actually capable of doing. We've implemented a lot of other algorithms that I won't go into great detail about today. So how do you actually do machine learning on knowledge graphs once you've actually built them? So it shakes out like this. You can basically do this in 3 different ways. There are matrix factorization-based methods, which we don't really have any implementations of, random walk-based methods, of which NodeDevec is one, and neural network-based methods. But all of them, more or less, will get you from a knowledge graph, obviously in graph form, and transform each node into this embedding space, this low-dimensional space, where its position in this space sort of represents all the information that you have about that node and its relationship to all the other ones. So things that are closely related in the network over here will end up in the embedding space close to all their neighbors in the graph. And so this is a kind of a convenient way of transforming all the knowledge that you have in graph form into this sort of lower dimensional Euclidean space that you can use for GraphML and other things. So this is not a particularly huge knowledge graph, but it's big enough to where it exposed some performance issues. So KG COVID-19 is about 500,000 nodes, and so our use case, as I said, was to find drug repurposing candidates. There are other knowledge graphs that we have made or been involved with the projects for making. Monarch is a knowledge graph for rare diseases. That's about an order of magnitude bigger, if not more. And these graphs, obviously, so we built them to take in the most recent data every month. And so it's sort of axiomatic that they're only going to get bigger because there's more and more data that's produced to go into them every month. And at least one of the ways you can do this is using some software called Network X to read in the graph. That was a bit too slow for our purposes. We looked at other more performant implementations of graph libraries and the algorithms that I just described. And those are a fair bit more performant. I mean, they were written to be more performant. They're written in C++ or some other performant compiled language. But even those really don't allow practical analysis of huge graphs. And so we were interested in sort of addressing that problem before we moved on to our use case. So we made in Smallin. This is as I said, a graph library, and this is implemented in Rust. What I'm showing you here is sort of benchmark of in Smallin versus Network X, which is not nearly as fast as and Smolin. I'm showing you the time requirements here for 3 different knowledge graphs, Monarch, KG COVID-19, and Phenolator is another biomedical knowledge graph I didn't mention, constructed by Tiffany Callahan at the University of Colorado. So you can see basically the time requirements of in Smolin versus Network X are about an order of magnitude less. So it's about an order of magnitude faster than Network X. And the memory footprint is also about 2 orders of magnitude lower, which is obviously a good thing if you're trying to deal with very large knowledge graphs. So that's actually just reading the knowledge graph in using the graph library. To do node-to-vec, you have to do random walks on the knowledge graph, which is also obviously very compute intensive. I won't go into great detail. Sorry, the legend in the axes are kind of small here, but I'll call your attention to the blue line. That's in Smolin. Sort of the competing libraries are, again, about an order of magnitude slower in loading the graph, and that's in terms of time. The memory requirements are also about 1 or 2 orders of magnitude higher for the competing libraries. The walks are much faster for in Smolin. These are first order walks, so in other words, not biased in any way. These are second order walks where the walk is actually biased towards either breadth first search or depth first search, basically. And so you'll notice these skulls mean when the other libraries actually died trying to process these libraries. We can comfortably, so you probably can't see this, but this is on the order of 100 million nodes. So we're comfortably scaling to hundreds of millions of nodes, and it's probably practical also to go into the billions of nodes here. So we're definitely performant enough to deal with KG COVID-19 at this point. So that's reading in the graph. How do we actually do graph ML? So in Smolin takes care of loading the graph and there's a lot of utility functions too that I won't go into. And Biggin, as I said, is built on Smolin and actually uses TensorFlow, which is some software from Google to actually do the graph ML. Node-to-vec is the algorithm I'm going to talk about, but there's, like I said, we've implemented, several other algorithms that we intend to use in the coming months and years for this sort of work. Okay, so how do you actually do drug repurposing using KG COVID-19 by applying knowledge graphs? So we start with the knowledge graphs and those sort of in a stylized way showed you the nature of the data. Here's SARS-CoV-2. There's actually a node that represents SARS-CoV-2. And there's tens of thousands of drugs. I've indicated here in green. We apply node-to-vec and, as I said, transform them into a low-dimensional space. And so conceptually, we end up with node-to-vec and some in the drugs surrounding it by some distance. And so what you can do is something fairly simple and say that, how far away is this drug to SARS-CoV-2 as measured by cosine similarity, which is basically sort of a way of measuring how close they are in latent space and how close is this one and this one. So this implies sort of a ranking of how close, how involved every drug is with SARS-CoV-2, and therefore how interesting it might be as a drug repurposing candidate. You can also do more elaborate things using the embeddings. You can train a neural network to sort of tell you what links to do link prediction essentially for the same purpose. So give me a score that this is a good drug repurposing candidate for SARS-CoV-2. So if you look at, for example, cosine similarity of all the drugs that we know about versus SARS-CoV-2, you see this nice Gaussian distribution, which is we assume just some random similarity. And then you see this very interesting shoulder off to the right. And these obviously are the ones we're going to be more interested in for COVID-19 drug repurposing. And just to give you an idea of the flavor of drugs that come out here, you're seeing a lot of interesting hits here, like steroids, among which is dexamethasone and prednisone. Dexamethasone is actually has been used for COVID-19 treatment. So that's gratifying and gives you some confidence that these are useful repurposing candidates. Antivirals also come up in the top 100. Antibiotics, strangely, because I say strangely because these are directed against bacteria, not viruses. We're not really sure what the signal is about. This was particularly interesting to us. We saw a lot of NSAIDs and COX inhibitors. These are things like aspirin, ibuprofen, and acetaminophen. These are the things that people tend to take without any hesitation when you have a cold or flu or COVID-19 or any sort of ailment like that. And so this we thought was very interesting because we wanted to know, whether or not this had an effect on COVID-19 outcome, and if so, what was the nature of that effect? Does it make it better or worse? So I'll show you some data about that in a minute. Okay, so this gives us sort of a ranked list of drugs that might be interesting to look at for COVID-19 drug repurposing. So how do you decide? And you obviously can't just go to a pharmacy and start handing these out to COVID-19 patients. We want to know if they actually worked. Is there some orthogonal way we can corroborate these or validate these lists? And so, like I said, the machine learning, for better or worse, will always give you an answer. And so we need some sort of orthogonal way of deciding whether or not any given drug is a good candidate. So for machine learning on biomedical data, an obvious place you can look to do this is using EHRs and medical records. So we developed a bit of expertise doing observational studies on EHR data to see what the effect of any drug is on COVID-19 outcome. And the way you do that conceptually is basically by doing this. Sorry, I ignore the top panel here. The bottom panel is explaining how retrospective observational studies work. So we start with COVID-19 positive patients. And for every drug, we do the following. We'll find patients that have an indication for the drug. So say, for example, this is a migraine drug we're looking at. These will be patients with an indication for that drug, patients with migraines. And so we'll separate them into two groups, those that were not treated with the migraine drug of interest and those that were treated. And then we sort of demographically align these patients with these patients. So this is called propensity matching. And the idea is to sort of mock a random clinical trial sort of after the fact. And then we can just compare the COVID-19 severity of this group with this group. So if this drug, for example, is having a positive effect, these patients will fare better in COVID-19 than these patients. And so if we look, going back to that sort of right shoulder here, what do these if we take the top 100 drugs, do we actually see a signal? So I'm showing you the p-value of an ordinal logistic regression, basically a measure of whether or not there's a statistically significant effect of those drugs, of the people that were on the drugs in COVID-19 outcome versus the people that were not on that drug. You can see a signal here. Down means a significant signal. 18 of the top 100 drugs have a p-value under e⁻3. And like I said, several drugs in the top 100 were NSAIDs and COX inhibitors. I'm not sure which is which here, but at least 3, and I think more than that were in the top 100 were COX inhibitors. This was particularly interesting to us, and so because, like I said, these are drugs that people take without any hesitation when they get COVID-19 or any other cold and flu, and so this is very much actionable knowledge to know whether or not this is having a beneficial or detrimental effect on COVID-19 outcome. And so here are the sort of results boiled down for the observational study on NSAIDs. NSAIDs are a kind of sort of a subclass of COX inhibitors. And they actually do affect COVID-19 outcome. Well, I should say they are associated with a better COVID-19 outcome. So if you compare the NSAID cohort versus the control cohort, you can see the incidence of severe or death from COVID-19 is less than in the control group. And likewise, invasive ventilation, AKI, this is acute kidney injury, and ECMO. These are basically detrimental. These are bad outcomes for COVID-19 patients. And you can see that they're much less common in the NSAID cohort versus control cohort. Another way to measure this is with odds ratios. So what are the odds that the NSAID group is going to fare better than the control group in COVID-19 severity, mortality, invasive ventilation, AKI, and ECMO? All of these are, the odds ratio is below 1, so they're doing better. These are the confidence intervals. So the long and the short is using, the actual information here is that NSAID use among COVID-19 patients is associated with a better outcome. So this gives you confidence that it's okay for, and in fact, it's probably beneficial that these patients are taking NSAIDs for COVID-19. So that's, I'll just summarize what I've told you here. So KG COVID-19 is obviously our COVID-19 knowledge graph. Here's a paper describing this work. The code and some documentation is here. Everybody's free to use the KG for whatever they like. This, as I said, sort of morphed into KG Hub, which we're working on to sort of develop this ecosystem for making knowledge graphs to address other problems in biomedicine. So we've used GraphML for drug repurposing. There's a preprint here. This is in peer review, and hopefully will be published this year. And this is sort of, I think, an example of a pattern that we can apply to other problems in biomedicine. So that conceptually, our formula for using knowledge graphs to address problems in biomedicine is to make a knowledge graph, apply GraphML, and then do observational studies in EHR, and as a way of extracting actionable information from knowledge graphs and actually validating them with real world data. Just let me say some quick thank yous to everybody. Of course, Chris Mungall is instrumental in all this work. This is more or less the entirety of Bebop, which is our group in Berkeley Lab. Peter Robinson's lab at Jackson Lab has been instrumental in all of this, and also Hannah Blau and Ben Coleman in Peter Robinson's lab. Tommy and Luca really took the lead in developing this performant GraphML software, and Melissa Handel, University of Colorado, the entirety of N3C, the N3C community. This is hundreds of people that really came together very quickly to help with the COVID-19 response in the US, and also in outside the US. I think with that, I will stop and answer any questions.
Fantastic. Wonderful talk.
Thank you.
I have questions, but others will have to.
And we have some wonderful things on the chat already. I do have a question for Justin and Chris, just quickly. You mentioned the possible use of some ontology design patterns that might be relevant for other aspects of this work. I know you had a slide on that. I wonder if you could say a few more words about that.
So you're asking about ontology design patterns, or more just kind of how ontologies are kind of like used in here?
I guess used in here, Justin raised the possibility that you might have some patterns to suggest that might be used elsewhere. And I was just wondering if you could give a little bit of a flavor of that.
Yeah, so I mean, so we make use of a wide range of ontologies here. So these ontologies are often built using structured design patterns. But in the ontology, we only put the very generalizable knowledge that is not non-contextual, kind of always true. So for example, in our disease ontology, Mondo, we'll maybe have an axiom that says something like every instance of COVID-19 is caused by infection with some instance of SARS-CoV-2. So we've got separate nodes for the disease and for the infectious agent. And this is a design pattern that we implement for all of the infectious diseases. So it sounds kind of trivial, but it's very useful to have these links connecting from the disease ontology into the ontology of organismal and viral classifications, because they don't always kind of quite align. So there's these kind of design patterns at the ontology level that we incorporate, but we also try and bring in kind of, one thing we want to avoid, one thing that's very tempting with knowledge graphs is that you can connect anything to anything. And so, I can connect my genes to people and people to infectious agents and so on, but that can lead to a lot of, it's suboptimal for reasoning with, because you've got to kind of like normalize the different ways of saying the same thing. So we make use of essentially kind of like design patterns / a data model for structuring our knowledge as well. And so that comes in the form of the BioLink model, which is, it's kind of like an ontology, but it's more, should think of it more like something like a kind of shaped language like Shackle, or a kind of more standard data model. And this constrains the kinds of ways in which you can relate things together. So the model tells you that you're allowed to relate diseases to infectious agents or diseases to phenotypes, but maybe not, you don't link the phenotypes directly to the infectious agent. They're always mediated through the disease, for example. And so there's a kind of interesting connection between what people are doing in the ontology design pattern business, which is more about structuring ontologies, and what people are doing with shape languages like Shackle, which kind of came late in the game, I think, for the semantic web, after OWL and so on. People realized they needed to kind of have more kind of closed world structures over their ontologies, and just more traditional kind of like data modeling, like UML type data modeling as well. So to me, these things all are encompassed under the general kind of heading of design patterns / data modeling. And they turn out to be crucial when building these large knowledge graphs from different sources, where you really want to try and impose some structure and some regularity on how knowledge is represented, and you don't just connect everything to everything with some kind of like big crazy hairball.
Thank you. That's very helpful. Justin pointed out that one of the attractions of the knowledge graph is, you essentially, it's a very simple structure, and it allows all kinds of things to be represented. But on the other hand, as you pointed out, you sort of want to structure certain things because you actually know that represents reality, and it will be more efficient. So having a best practice by which you take, starting with great flexibility, but then you structure it in a meaningful way, modestly, it really is the way we want to go, I think.
Yeah. And one of the things we just went over quite quickly there was just the huge amount of effort is to do the ETL here. So we've got all of these fantastic databases in the life sciences about drugs and chemicals and so on, but they all store things using their own bespoke file formats, bespoke data structure. So there's a lot of work in just building the code that will slurp data from these different sources, keep up to date with how they change their schemas, and map these into our common data model.
Have you had to change the structure much? I'm sorry. Have you had to change the structure much going forward? Have you realized new things? So you essentially have added structure or changed structure in some way that's significant?
Can you think of a good example of that, Justin?
I'd say we've gotten better at normalizing IDs. It can be challenging to make sure that remdesivir, for example, from this database is, ID normalization is. That's kind of there's all these long tail of issues with that kind of thing. So that's been refined, I think, over the last year. Really, the main lift as far as ATL, once you've done it, it's not trivial to do it first, but then it's always this Sisyphusian labor to keep fixing it when people move URLs, or they change the upstream file format changes, or they just get rid of one column that we need, or something like that. That's the main challenge, I would say. The structure, I would say, has not changed wildly over the last year or two.
One of the reasons I ask is that, science discovers new things all the time. And the question is, in this difficult area of pandemics, COVID, whether science has discovered something that we didn't start with, and now we know, and therefore it's reflected in the knowledge graph.
I mean, I think definitely we see the changes over time in the actual knowledge represented itself, but the knowledge structures, the data model is so high level. We don't really see a kind of a new scientific discovery overturning, just our broad kind of structures, some of the kind of structuring changes that do occur over time are, as Justin says, things to do with ID normalization and choices about whether to collapse or conflate two different entities. Like, do we have separate nodes for a gene and the product of that gene, which is typically a protein, when most of the time for our purposes, these things are one-to-one. We sometimes make a decision to have separate, concepts for these, but sometimes we merge them into one. But that tends to be more driven by, just pragmatic concerns rather than, reflecting a new scientific discovery or something like that.
Yeah, the type of thing I was thinking of is a discovery of a mechanism by which the cocks work on this. Maybe there's something that we don't know about a mechanism there that might be discovered because you see the effect, but you don't know why.
Mm-hmm.
This is sort of maybe a tangent, but a related point is something we're interested in doing with all of these knowledge graphs is you can apply, so we made a sort of a GraphML model to extract knowledge to do with NSAIDs and their relationship to SARS-CoV-2. But what you can sort of you can describe that machine learning task in YAML format and then just sort of keep doing that every time there's a new build. So on the subject of sort of finding new knowledge as it occurs, we think that's kind of an interesting way to sort of automatically update, as with a new build, new knowledge is going into the KG, and so you can sort of automatically build that into the KG, automatically apply the GraphML model that you applied last year and see what shows up, what's new this month, so to speak. So it's kind of like a self-updating GraphML model. So we're working on sort of operationalizing that now.
Ravi has his hand up for a question.
I have a question. I have several, but that's why I wanted others to go first. One of them is related to the otolaryngological ecosystem. There are multiple bacteria or active agents that affect our health, the oral health, etc. And it seems COVID has found a favorite space in the nose, as everyone knows. So how much does this virus affect other correlated bacteria of the ENT area? And is that important for drug treatment? And second question is a little more broader, which is how do you deal with correlative studies, correlations, for example, among drugs and the genes?
Can you explain the latter question?
The latter question is how these knowledge graphs are allowing you to study correlations, the strength of statistical correlations rather than binary relationships. Data models give you binary kind of relationships in queries.
Yeah,
more statistical.
Yeah, so this is something I think we'd like to, I don't know if you can talk, Justin, about or any latest efforts here, but we'd like to, even though most of the knowledge we have is kind of like, binary, A binds with B, for many of these edges, we can associate weights with, like the protein-protein interaction. We have kind of like, confidence scores and weights and drug binding, there's kind of like, there's various kind of like, measures of, how coupled they are. I guess we're not quite using those quantitative measures in the KG approach yet, but I think there's some approaches that we're looking to make use of there.
Yeah, that is kind of an interesting challenge because, like, for example, string and intact, those are our PPI sources. They will give you a floating point number, of how strong or how likely that interaction is. But the other interactions, there are other data don't give you a number for that interaction or whatever that edge is. And so it's not 100% clear how to harmonize those things, but I think we have some ideas about how to do that. I don't think we have done that thus far. It's an interesting idea. As far as the sort of microbiome question, I think there's a subgroup in N3C that is working on that. It seems very likely that there are that the microbiome context in which all this is happening is likely to have an effect on COVID-19. It's not entirely clear what that relationship is, and it's probably a bit outside my area of expertise. But yeah.
It would be, for example, thank you, Chris. It would be, for example, Chris and Justin both. It would be interesting to see how COVID is helped by other biomes in propagation to pulmonary area, for example, etc. Why certain types go faster in the lungs than others, etc. I'm just giving some examples. But you have done tremendous amount of integration work in your papers, in your work, in your collaborations. It's just phenomenal. If you asked a scientist of 10 years ago, even when GEO was known, they would not be able to think of this kind of comprehension of collaboration. It's fantastic. And I do have also that question, why is that one peak in that cosine graph? That cosine graph, one peak. Why only one peak?
I can't prove this, but I think what you're seeing here is just sort of random distance from SARS-CoV-2. The one peak I think is just
is it mathematics or is it more than that?
I don't think it's a sort of a mathematical artifact or anything like that. I think these are just, well, it depends what you mean by what I would mean by artifact, but all of this, essentially this entire knowledge graph is occurring in one sort of connected component. And I think that there is a bit of bias in these and other knowledge graphs where, you have these sort of star nodes where like everything's connected to them. So this could be drugs that are closer to that star node than others. I think it's more likely though that these are just drugs that are involved in an important pathway in SARS-CoV-2 or otherwise just close to, more than closer than a few hops to SARS-CoV-2.
What is the horizontal scale?
This here is cosine similarity. So this is 0. This is 1.
Okay. Thank you.
Sure.
I know that on chat we have a lot of questions. One from Mike Bennett.
Thanks, Robbie. Yeah, Chris has partly answered it, but I'm just curious. You're talking about, you're sort of living in the world of data, and it's all about the data, and there's always a relationship between data and the things the data is about. And since some of those things are, ears and noses and throats and lungs and so on, and viruses, have you had to deal with the relationship between the data of the things, or stand up ontological representation of things that are not maybe in data, and have you dealt with that kind of duality?
I think that maybe
one way to think about it is, we do tend to focus on the knowledge in the knowledge graph rather than the raw data, but then we take this knowledge graph and apply it to the raw data. So, the canonical example there would be the patient data from N3C, and we use the KG to essentially help us interpret that data. But I think we do need more mechanisms to more flow between the two, such that, you know, if there is raw data being generated, whether it's kind of like epidemiological or gene expression data, just a kind of clean path to kind of like lift that up into kind of simple triples that can be expressed within the KG.
I have a favorite question. That is, our chair of the Ontolog board has developed a query language called KGSQL. How could that be useful in your work? I would like a short dialogue, if possible, between Ken Baklowski and you. Ken?
Yeah, I don't know that you could make use of it right now because there isn't any tooling available. It's just more in the way of a proposal. But the idea is that the edges in your graph will be first class, so they could themselves be annotated. So this would allow one to have a better idea of the provenance of the data. And actually, that was one of my questions, was how much does this could you comment on explainability? So I mean, you find some association, something that's near in your space. You might want to go back and say, well, why?
Yep. I think that this is one of the many attractive things of doing GraphML and KGs is that the explainability in some way arguably is built into the knowledge graph. So if you have SARS-CoV-2 and some NSAID, you can, for example, interrogate the KG and ask for the 10 shortest paths, say, between those two, and then just look at them. And that in some way is explainability sort of right out of the box. Which of those, I mean, because that's if you apply node to vec, for example, those top 10 are a part and parcel of the reason why it's coming up as a hit, so to speak. And so I think this is kind of where knowledge graphs shine with respect to GraphML.
Yeah, just to, our data model as well that we use, I like this idea that you said, Mike, of, treating the edges as first class entities. And we do, even though they're not used in the ML, and they can still be used for explainability, we include provenance on each of the edges. We say this came from this trusted source, from this PubMed ID, and so on. So, we're as we kind of progress, we're kind of putting more and more information on those edges, rather than kind of inflating the number of predicates that we have. We kind of put more qualifiers on the edges. We're still looking at methods to kind of like really exploit some of those, but we're definitely be interested in query languages that allow you to do that. I mean, we do have a triple store instance of this, and you can query it, but we use the kind of like the standard RDF reification model for doing that, and it can be quite awkward to query edge properties using RDF reification. There is this kind of effort in the semantic web world right now moving towards RDF star, but which is a step forward in terms of like treating edges as first class, but there's still limitations in using it. And Ora Lasilla, one of the original authors on the semantic web paper with Tim Berners-Lee, along with Brian Thompson, who developed BlazeGraph, have come up with this 1G graph model that kind of is kind of essentially a simplification of RDF, but at the same time treats edges as first class. And you might want to look at that for a potential target for KG SQL. So
yeah, well, the idea of KG SQL was to have an improvement on KG, the RDF star approach, which if you look at my article or the talk that I gave in this forum, you would see that there are a lot of disadvantages to using reification. That now the reified fact isn't the same as the fact. Different things. Whereas in KGSQL, they're the same.
Yes. Mhm.
So it simplifies your queries. It's some, much more, a much simpler really approach to this kind of annotation. But as I said, I mean, at the moment, there are no tools, so it would be difficult to use KGSQL as it is right now.
Right.
Yeah,
I'm
thanks for your
going to have to
comments.
head to another call, but I can leave you chatting if you like.
Thank you
But yeah,
so much. Thank you so much. I guess we are very close to the end. Are there any burning questions? Otherwise, I would request that we follow this on with more email threads and questions. This is very, very interesting talk. We really want to thank you so much.
Thank you all.
Yeah, thank you.
Thank you for coming. It's one of
Thanks,
the.
Chris. Thanks, Justin.
Yeah.
Thank you very much.
Travi.
Thank you so much. Bye now.